All right, so today we're diving into something that sounds, I know, incredibly complex. C9orf72. But I want you to think of it like this. It's a glitch in our genetic code. A single tiny typo in a massive instruction manual that ends up causing these absolutely devastating problems. It's a story about a biological puzzle, a high-stakes detective hunt, and honestly, a revolution in how we understand the brain. So, let's get into it.
And this whole story, it really kicks off with a mystery. For years, scientists were just stumped. They saw this bizarre connection between two diseases that, on the surface seemed to attack our bodies in completely different ways. How could they possibly be linked? How could they be partners in crime?
So picture this. It's the early 2000s, and researchers are basically dealing with two totally different villains. On one side, you've got a disease that's attacking the body's ability to move. And on the other, you have a disease that attacks someone's personality, their behavior. And yet they kept seeing them show up together in the same families. Okay, so let's break that down. On one hand, you've got amyotrophic lateral sclerosis, or ALS. This is a disease of the motor neurons. It affects physical control. Then, on the other hand, you have frontotemporal dementia, or FTD. This one attacks the brain's frontal and temporal lobes, literally changing the very core of who a person is. They seemed completely separate, right? But time and time again, both diseases would pop up in the same family trees. It was a huge clue that a single hidden culprit had to be at work. Well, this puzzle kicked off a massive scientific manhunt. Seriously, finding the shared genetic link wasn't just about lab work. It was a genuine detective story. You had scientists all over the world following these tiny clues hidden deep, deep inside our DNA. The first major break in the case came in 2006. Genetic mapping pointed all the detectives to one suspicious neighborhood, a specific region on chromosome 9. For 5 long years, scientists scoured that area until 2011, when, boom, 2 independent teams at the same time finally cornered the culprit. They'd found the glitch. It was inside a gene they named C9orf72. So what was this glitch? It wasn't just a simple typo. It's something called a hexanucleotide repeat expansion. What that means in plain English is that a small chunk of DNA code, this little sequence, GGG, it just got stuck on repeat. Instead of being written a few times like it's supposed to, it was copied over and over and over, like a song stuck on a loop. Now, to put that in perspective, in a healthy person, this little GGGCC sequence repeats a handful of times, maybe somewhere between 2 and 30. This is completely normal. It's the baseline for how the gene is supposed to look. But in patients with this specific kind of ALS and FTD, that number just explodes. It can repeat hundreds, or in some cases, even thousands of times. I mean, imagine a recipe that says add a pinch of salt, but a printing error makes it say add salt 1,000 times. The end result is going to be catastrophic. And the significance of finding this one mutation? It was absolutely staggering. This single genetic typo accounts for a whopping 40% of all familial, that is inherited, cases of ALS. Let me be clear about that. It's not just a cause. It is the single biggest, most common genetic culprit we know of, period. Okay, so we found the glitch. But the next big question was, how? How does a simple genetic stutter cause so much devastating damage? Well, it turns out this one mutation is a triple threat. It attacks our cells in 3 distinct ways, all at the same time. So this one glitch wages a 3-front war on our cells. First, the cell itself struggles to read the stuttering gene, so it ends up making way less of the normal C9orf72 protein. Think of that protein as a cellular traffic cop, and when it disappears, things get messy. Second, the faulty genetic message itself, the RNA, it forms these sticky, toxic clumps. It's like throwing gum into the gears of the cellular machinery. It just jams everything up. And third, and this one is just bizarre, the cell actually creates brand new toxic proteins from all those repeats. They're like defective little robots that just run around poisoning the very neurons they were created in. The fallout from this triple threat, it's just immense. Neurons get totally stressed out as their power plants and supply lines fail. Those toxic rogue proteins can actually spread from one cell to the next, almost like a virus. Then the body's own immune system gets confused by all the chaos, and it goes into overdrive, mistakenly attacking its own neurons. And because this damage hits the frontal and temporal lobes so hard, you get the FTD symptoms, those profound changes in personality, right alongside the physical decline of ALS. But here's the powerful turn in the story. Finding this gene wasn't just about identifying a cause. It was like finding a map in a dark forest. For the very first time, it gave researchers clear targets. It opened up entirely new paths forward in the fight.
You have to understand, before 2011, research into ALS and FTD were happening in pretty separate worlds. This discovery shattered the wall between them. It connected these two fields, allowing scientists to pool their knowledge, to share data, and to collaborate in ways they just never had before. It was a complete paradigm shift.
A whole new playbook for research just opened up overnight. Scientists could start designing therapies, called ASOs, that act like molecular scissors to go in and snip that toxic RNA message before it can do harm. They could start looking for the toxic byproducts in spinal fluid, using them as fingerprints to track the disease. And incredibly, they could even grow mini brains in the lab from patient stem cells, letting them test potential drugs without ever putting a person at risk. So where are we now, nearly a decade and a half after that discovery? Well, the progress has been truly remarkable. Now the fight is far from over, but there is genuine, tangible hope on the horizon. And this progress is real. You can see it. Those ASO drugs we talked about, they're now in human clinical trials, and early results show they can cut down the toxic RNA by as much as 70%. In the lab, gene editing tools like CRISPR have already successfully delayed symptoms in animal models. And those biomarkers, they're not theoretical anymore. They are being used right now to track the disease in people and to measure if new treatments are actually working. Now, let's be really clear. These are not cures, not yet, but they represent real, meaningful progress. We are finally moving from just managing symptoms to directly targeting the root cause of the disease. The path that was once so dark is now lit. And that kind of leaves us with a final big thought. The story of C9orf72 is incredible because it shows how cracking a single genetic code can completely revolutionize our understanding of the brain. It solved one mystery, but in doing so, it gave us a key. So the question now is, what other locked doors will this discovery help us open?